imagine some Turing machine or something at the level of molecules that's operating on DNA. But such a machine would be so complex that it's hard to imagine that we wouldn't have stumbled upon it. I mean, this is a Turing machine that operates on DNA would be more complex than, let's say, a ribosome, and ribosomes are visible in electron microscopy. So why haven't we seen anything like this? So this is what I'm going to try to propose today, that there is a system that is simple enough that you can imagine it being have gone undetected in the decades of molecular biology, but is also a universal computation system.
So welcome back, everyone. Today we are joined by Hessam Akhlaghpour. I only got one chance to practice that. I hope I did okay. So Hessam is a postdoc at Rockefeller University, and he's working at the Maimon lab, I think on a lot of very different interesting topics, including neuroscience and some RNA biology, computational theory. And he's going to share one of his latest papers that came out in the Journal of Theoretical Biology called An RNA-Based Theory of Natural Universal Computation. And we're going to dive into it through a presentation and a discussion. So Hessam, will let you take it away. Thanks so much.
Sure. Thank you, Carlos and Perry, for having me. Do I share my screen right now? How do I? Should I share my screen? Or would you prefer? Sure. Okay, cool.
That works. Yeah.
It says the host disabled participant screen sharing. I'm waiting for.
My bad.
These are rookie mistakes by Carlos right now.
Yeah, honestly, it's been a while since I make this kind of blunder. Let me go. It's okay. We don't have a time limit, so we can just click around. Okay, you should be good to go.
Hessam, you're muted. I don't know if.
Oh yeah. Okay. Okay. You can. I'm assuming you can see my screen and my cursor moving around. Okay. Perfect. All right. So, hi everyone, and thanks for having me present this paper. I'm going to present some theoretical work that attempts to answer the following question: where is life's universal computer? And this is not a question that's been raised or even recognized as a serious scientific question really in the literature before. So in the first part of my talk, I'm going to try to clarify what I mean by universal computer. And I'm going to motivate that question. And hopefully I'll convince you that it's a meaningful question that is worth investigating. And in the second part, I'm going to present to you a theoretical model that attempts to answer this question. And my primary goal is to show that universal computation is within the reach of biology at the molecular scale. And I use that to argue that an undiscovered RNA-based computation system might exist, the details of which might need to be discovered experimentally. But this is a proof of principle that it can exist. And personally, I'm really excited about raising this problem because I think the more people can get on board with trying to find this or solve this problem, the more likely it is that we will find it in my lifetime. Okay, so first, what do I mean by computation? So life needs computation for nearly everything, from when an insect needs to fly and use visual cues to guide flight, to things like human language acquisition, to even at the scale of single cell biology. This is an image of a stentor that has been shown to have hierarchical avoidance behavior that's pretty complex, and you usually expect this kind of stuff to only occur at the level of highly complex organisms, not single cells. And at the level of embryonic development, which is also a computation problem. If you think about it, you have to start from a single cell and build a highly complex three-dimensional structure and you have to do that through coordinating with daughter cells of that starting cell. Okay, so it is generally assumed that the building blocks of biological computation have been identified. And these building blocks include things like neurophysiology, neural networks, molecular signaling cascades, and genetic networks. So this last figure is a very, I guess, historical figure that people bring up a lot. This is Waddington's epigenetic landscape, where a single cell is conceived as being a single dot in a plane, and as this marble that you conceive of is rolling down a hill, this determines the cell's fate, and there you can get embryonic development and different cell types. Okay, so for these different kinds of computation that we see at different scales of biology, we think neurophysiology, neural network dynamics, and synaptic plasticity rules is involved at the level of cognition and learning. And at the level of single cell behavior and embryonic development, we think it's molecular transduction pathways, gene regulatory networks, and even non-neuronal electric signaling. And generally now the problem is, okay, let's find the parameters of these computation systems. There's no mystery in how computation can occur. It's just finding the right parameters, finding how these building blocks are used to construct different computation systems. Carlos, you go ahead.
Yeah, I have maybe a naive question, but is there, I think, I don't know if I'm remembering this from the paper or from the title of your blog, but your blog title is Life is Computation. Is there a distinction between life being computation and life using computation to solve certain problems?
Yeah, I mean, that kind of the title of my blog is kind of more of a maybe a poetic statement, not really a scientific statement. That kind of depends on how you choose to define life. I personally think that the most important aspect of life is computation, and so it makes a lot of sense to define it in terms of computation. But I mean, I can imagine someone else might disagree, and I don't have a problem with that. We can call whatever we want life, but computation to formalize it a little bit more than just an intuition. I think of it as any problem that involves the informational content of the input and output rather than the material content. So for example, can I replace whatever is doing the computation for flight with a microchip, like maybe a really small microchip? Can I replace that part in a ladybug's brain and have it fly? I guess so. Yeah, maybe there's, but I can't replace the digestive system of, animals with a microchip. So it's not, in some problems, it's not the informational content. Some problems it is.
I guess, yeah, there's maybe the way you think about computation, if you just think about it in the way that we just use computers every day, is there exists a problem and it's kind of outside of the computer in a way, and you kind of feed it through the computer. But when you think of it in the physical world, it's almost like it's one and the same, right? Like the butterfly flying is just almost just following some mechanical rules that are, it looks like maybe you can say it looks like it's solving a problem, but actually it's just following a deterministic process. And then the idea of a problem might be some kind of abstraction. But I mean, I haven't spent very much time thinking about it, but yeah, I wonder if there's some kind of definition there that might need to be worked on.
Yeah, and actually that's a good segue to the next slide of what is computation. So basically, what I'm going to be trying to argue is that we've been neglecting a lot of the insights from the theory of computation, and we haven't really been taking it seriously. The kind of study of computation in biology would benefit from bringing in these insights and taking them more seriously. So in the theory of computation, a problem is defined as a function from an input domain to an output domain. So for example, the problem of squaring a number is a function of integers to integers. And it doesn't have to be, it can be a partial function. Maybe some inputs don't produce an output or whatever, that's fine. And a computation system is a system that can solve multiple functions. So for example, let's say neural networks is a computation system. You can use it to solve many different problems. And one way to formalize this is say, every computation system given a descriptor maps to a function, an input-output function. So that descriptor can be, for example, in the case of neural networks, it could be the set of weights and activation functions and all the details of who's connected to who. That would be your descriptor, and that by itself defines what the input-output function is that it's going to solve. Okay, so then you can ask this question of, okay, what's the scope of functions that a computation system can solve? What are the set of functions that, let's say, neural networks can solve, or maybe Turing machines can solve, or a programming language like C can solve? Well, there's some important insights from the theory of computation and that's that there are different scopes of computation. And this is just, I guess, a subset of some of these computation systems that have equivalent computation power. So let me just start from the center one and move our way out. The weakest computation system that I've depicted here are lookup tables or combinatorial logic circuits or basic decision trees. And all three of these systems are equivalent in the sense that any problem that can be solved by one can be solved by the other, or you can use let's say a combinatorial logic circuit that's made of AND gates and NAND gates and whatever to implement a lookup table, and vice versa. And then as you move your way out, you get to more powerful computation systems like finite state machines, which are machines that can take a string as an input and change the state as it receives every new symbol. And then at the outermost layer, you have universal computation systems, and the most popularized of these systems are Turing machines. They're equivalent to non-deterministic Turing machines, quantum computers, and all of these systems can be used to solve, to simulate any of the other systems in this list. And it's been shown, well, it's been, I guess, conjectured that this is the limit of computation. This is what you can compute with finite means. Any computable function can be computed by any of these systems here in this list. Okay, does that make sense? Now, okay, so what about biology? What's the scope of the models that we have for computation in biology? And there's a conjecture by Chris Moore from 1998 that kind of illuminates what we know about the computational scope of our models of biological computation. And in this conjecture, he says that generic finite dimensional dynamical systems are incapable of universal computation. That is, they don't fall into this outer layer. And all of our models of biological computation are encompassed in the set of generic finite dimensional dynamical systems. So if that's true, then maybe they're at this level of finite state machines and not outside of this. Go ahead, Carlos.
Would you be able to just give a little bit of insight on what you mean by a dynamical system, if you're not going to do it later?
Yeah, absolutely. Yeah, I'm not going to go into this later, so ask as much as you want right now. So a finite dimensional dynamical system is a system that has a predetermined number of variables. That is, before given the input, in the descriptor of that computation system, you have to determine how many variables that system includes. And the system is governed by a set of usually differential equations that determine the rate of change of every single one of its variables, dependent on as a function of the other variables. Now, by generic, what Moore meant was systems that have a non-zero volume in the space of all possible dynamical systems. To be more precise, what that means is if you imagine you can imagine every dynamical system as a point in the space of all possible dynamical systems, and if that point is what do you call it? If it's homomorphic, if it's homomorphic to all its neighboring dynamical systems, then it's considered to be structurally stable or generic. What this means is if I give you a unstable dynamical system and you add the smallest amount of change to any of its parameters, like change a parameter by 1 part per billion or something, then you're going to get a new dynamical system that doesn't resemble any of the properties of the previous one. And so that dynamical system is considered to be non-generic. You can't find it to occur in nature, and you can't implement it. You can't engineer it.
So it's like non-continuous in this dynamical system space, if you want.
Yeah, it's a point that has zero volume. So most of dynamical systems that we work with, pretty much every recurrent neural network that people use in machine learning is surrounded by a neighborhood of homomorphic dynamical systems. And so it's considered to be relevant to physics. You can find it to occur in nature, or you can implement it, you can engineer it. But the kind of dynamical systems that have been to date shown to be capable of universal computation, they lack this property of structural stability.
And this is not a theorem. This is just like empirical evidence.
Well, it's a conjecture, yeah. So there was, in the 90s, a bunch of people. I think the first person was Chris Moore, and then a bunch of other people had different versions of dynamical systems that are capable of universal computation that can simulate Turing machines. All of those had this property lacked the property of structural stability. And then in 1998, Moore put out this conjecture that said that generic finite dimensional dynamical systems, or structurally stable ones, are going to be incapable of universal computation. No one has proved it, so it's still standing today. It might be wrong, but if it's true, then we're out of luck with finite dimensional dynamical systems. Okay, does that make sense?
Thanks.
Okay, cool. All right. So and now let's think about what biology is capable of. So the idea that biological computation systems are just stuck down here, or stuck somewhere in, let's say, finite state machine land, and cannot reach universal computation is kind of, it strikes me as strange and hard to believe, particularly because it's very easy to stumble upon universality by accident. So people have come up with different mathematical systems that later have been proven to be capable of universal computation, capable of simulating Turing machines. And some notable examples are Wang tiles, Conway's game of life, and Wolfram's rule 110. All of these are systems that can simulate Turing machines and have the capability of being used to compute any computable function. Okay, and one important property that all of these systems share is that memory usage can grow. So after you're given an input, the amount of memory that your program is using can increase, as opposed to having it be predetermined by the descriptor. So in finite state machines, the descriptor determines the number of states that your system can have. And you're stuck with that no matter what input you're given. But in these systems, if you conceive of your program as, like, let's say, the subset of space that's that you call your program, that program can grow in if it's Conway's game of life, it can grow in this two-dimensional space and recruit more memory. Does that make sense? Okay. Yeah, go ahead, Carlos.
I don't know if this is looking at it from too far out, but where would some model of the human brain fall into this? I mean, I can imagine new links get formed by neurons and things like that. Is that something that you can deal with or put into this kind of hierarchy?
Yeah, so it depends on how you formalize your model of the brain. So you could come up with some neural network model that's different from the way we do it right now, where it has a set of neurons that can be used, an arbitrarily sized set of neurons that can be used as general purpose memory. And if you do that, then you might be able to achieve universal computation. Now, I don't know of any biologically plausible systems that do that, or any models that are intended to model how we actually think, or how biology actually computes, to have that property of having a set of an arbitrary sized set of neurons that are used as general purpose memory.
So does that mean, when you say arbitrarily sized, can that be an infinite size, or do we still assume it's finite?
Well, we assume it's finite.
Okay.
The same way that, okay, think of it this way. When you, like in our computers, when I write a code in C, it's that code's going to work regardless of what the size of your memory is on your computer. If you've run out of memory, you can put the same code on another computer with larger memory. In a sense, the memory capacity of that system is arbitrary. Of that computation system is arbitrary. You can just grow it, and then it's still going to function the same way, the intended way. Is there a neural network model that works like this? Usually, our neural network models, the way we formalize it, the descriptor includes all the neurons and all the weights and everything. If you want to achieve universal computation, my intuition is that you need a system where the descriptor is not all of the neurons. It includes a reservoir of neurons that can be used as general purpose memory, and then somehow control that general purpose memory for doing computation. And I don't know of a model that works that way.
So are you saying we are not so this is for neural networks? What about human beings?
Yeah, so that's a good question. I think human beings are almost certainly universal. Human thought, I think, is based on a universal computation system.
But we still have finite number of neurons in our brains. Then.
Yes, the same way that a computer has a finite amount of memory. But
okay,
the system,
go ahead. Sorry.
The way that you formalize computation for computers, let's say for programming languages, you assume an arbitrary sized memory, and that's why it works. That's why you're able to simulate an Turing machine, and that's why you can take, if your program runs out of memory, you can take the same program without changing the code. You can take the same program on another computer with larger memory, with a higher memory capacity, and then it'll work. You can't. That's not how. Like, let's say if a neural network hits its capacity, and however you define capacity of a neural network, you can't take the same descriptor and put it in another network, and then it'll just work. So that's the distinction.
Yeah, thanks for clarifying.
Sure.
I guess I don't know if this is the slip up, but just because you can recruit more memory doesn't mean you're eventually not going to be able to do that, because you'll reach the global limit of available memory in your environment, right? And then, I mean, it's probably a definition thing, but I guess that's where I'm kind of slipping up.
I guess it's in the descriptor, right? If your descriptor does not specify the memory, not need to specify the memory, then you have a universal quantum computer.
Yeah.
I don't know if that makes, I don't know if what I said is true. Hessam.
No, that makes sense. Yeah.
But I
Okay,
guess you could just give your descriptor all the possible memory. No. Upfront.
But how would you do that? Would you give it an infinite size like set of neurons?
Yeah, I mean,
Yeah. Yeah. Go ahead.
I guess in any physical system, we're going to have a finite amount of resources. So if universal computation is achieved, it has to be in a setting where the total amount of memory is finite, no?
That's right, but it is abstracted out of your rules.
Yeah,
So,
I see.
and the same issue happens with finite state automata. People think, I think incorrectly, people think of Turing machines as infinite, and then finite state machines as finite. But the problem with that is with finite state machines, the advantage of finite state machines is they can deal with unbounded input size. So in finite state machines, you also have, you're assuming infinite time or unbounded time, not infinite. You're assuming unbounded time. If there's going to be an input for which a physical implementation of a finite state machine will fail, it'll decay or run out of battery or something, and then you can say, okay, well, it failed. I'm going to take the same descriptor for my finite state machine and run it with something with more a time and more battery or whatever, and then it'll work. The same issue happens for weaker computation systems. It's not just a problem with the model of Turing machines. Okay, cool. I'm gonna go ahead. So all right, so hopefully this is enough to justify the question. So first of all, physical systems, including biological systems, can be analyzed in the framework of computation theory. Now, I didn't go into this, but there is some valuable work that has been done recently that formalizes how we can compare computation systems that work on very different domains. So, in computation theory, everything's from numbers to number, integers to integers, or maybe strings to strings. But someone might come up and say, okay, well, we're not working with strings in biology. But there are formalized methods for how to compare systems that work on very different domains, and we can get into that if you want. The second point is that the universal scope is a mathematical upper bound to the scope of effective computation, and a universal computing device would immensely benefit organisms that struggle to survive and reproduce. And we know that evolution is capable of designing very impressively complex systems according to principles of optics, like your eye, mechanics, chemistry, physics. So why can't it evolve something based on the principles of computation? And finally, universal computation is easily attainable with very simple rules, as we've seen in the past 100 years. People have stumbled upon it by accident through systems that weren't really intended to be universal computation systems. Yeah, go ahead, Carlos.
So maybe we're jumping to discussion here, but I guess, do you think that computational universality is a feature that natural selection can act upon? Because I guess the other ones, like optics, can you see or not, is something that you can obviously directly link to survival. But this idea of the computational capacity of something, I guess it's hard to connect that to natural selection or fitness in an environment. Are there examples where that link can be made?
Yeah, well, I guess I can come up with stories of how it can be made. I don't know how useful that would be. In my intuition, it's just so easily achievable, and obviously more advantageous than weaker computation systems. And we see that in the development of human technology too. Like we have systems that could have been okay without a microprocessor, but we're putting microprocessor in them anyway, toasters or microwaves. They're probably even more efficient with these microprocessors and with a digital computation system doing what they want to do.
So, so in this example though, wouldn't it be better? I'm just thinking, like for a toaster, maybe it would be better to optimize fully a lesser computational system for the toaster and every system on the planet, than have a universal quantum computer, universal computer in each of them. But we just run out of time, I guess, in the lifespan of our universe. So we just say we have something that's good enough for everything, and let's put it in everything.
Yeah, but that's assuming that there's a cost for putting in this universal computer. Like it's hard to maintain or hard to discover or something. So we're gonna go with a more weaker computation system that fulfills the needs of a toaster. But that's that's not the case. At least with technology, we know it's not the case. It's more efficient. It's
So if I fully optimized the toaster computation, like have the fully optimized, you're saying it would be equivalent to just having a universal computer in the toaster.
Well, I'm not sure what you mean by optimized. Like, I guess you're saying if you come up with some analog, version of a toaster, and just optimize, like, let's say maybe it's energy or stability or
like
something.
it does this specific task extremely well.
Yeah.
I can think of, let's say, I don't know if you know Bitcoin miners, say, they're maybe not universal computers, but they hash extremely efficiently.
Yeah.
They compute one function extremely well, more efficiently in terms of energy and time, I guess, than my laptop, which is a universal computer.
Bitcoin miners are on computers that are run on let's say microprocessors, right? They are based on a universal computation system. See, it's not that a universal computation system is able to compute anything. You need to put in a program for it. Like this is not to say like any organism that has this universal computer can compute anything. The same way that my computer is right now not able to compute anything until I provide it with the appropriate program. It's just
I just thought the hardware. I thought the hardware was optimized, but maybe I'm not saying the right thing. I guess my question is, if I could optimize, say, the analog, maybe analog is not even the right word, but maybe I can optimize the toaster, the toasting process, without having access to all this universal computation. Is that still the same as just putting a universal computer in the toaster?
I'm not sure, because I guess it depends on what you're optimizing. I mean, I'm open to the idea that maybe there's some benefit to having an analog computer in there for some purpose, that you would, like, I guess you would bypass the microprocessor and just use an analog circuit for something. I'm open to that idea. I just my point is that it has so much utility, and we've seen that in human technology, and like I can come up with a bunch of different stories, but of how it can be used in biology. But I mean, just from the examples of where computation is useful in biology, from the four examples I gave at the beginning: embryonic development, or a cognition. These are all very complex tasks, and I can just see how it would be advantageous to have a universal computer sitting behind them and solving them. I actually think it's really hard to do without a universal computer. That's why I think it already exists.
Yeah, I guess maybe the notion in evolution that something that's not useful, even though it has a low cost to maintain, will degrade if it's not useful. It'll just decay with random mutation unless it actually is necessary for its current task. But yeah, I guess it could still be that it still exists, but we just don't see it. Or that is a feature that gives the system some level of like plasticity to changing environments, and it's kind of behind the scenes actually doing, actually helping the toaster sometimes have to do, I don't know, grilling instead of just toasting. Anyway, yeah, it's interesting stuff.
Yeah, yeah. Okay, so let me put in the last point that network models of computation are not capable of universal computation, which means we haven't found what could be life's universal computer. So putting all this together, the question comes up. Okay, so where is life's universal computer, if there is one? To go into the second part of this presentation, I'm going to start with a quote from Alan Turing in his 1936 paper. I'm going to read off of it. Computation is normally done by writing certain symbols on paper. I think that it will be agreed that the two-dimensional character of paper is no essential of computation. I assume then that computation is carried out on a one-dimensional paper, that is, on a tape divided into squares. I shall also suppose that the number of symbols which may be printed is finite. If we were to allow an infinity of symbols, then there would be symbols differing to an arbitrarily small extent. The effect of this restriction on the number of symbols is not very serious. It is always possible to use sequences of symbols in the place of a single symbol. And we see this in modern day computing. You can encode anything you want with just two symbols, 1 and 0. Now, this is obviously way before the discovery of DNA, and this could be thought of as a prediction of what DNA looks like. It's highly reminiscent of the structure of polynucleotides, DNA and RNA, where you have a fixed set of symbols, a finite alphabet, and from that you can compose arbitrary sequences of these symbols. And I have to say that one of the main influences of the way I think about this came from this book, Memory and the Computational Brain by Randy Gallistel and Adam King, which kind of allude to this idea that maybe computation is happening at the intracellular level inside neurons, and they involve polynucleotides. And Randy Gallistel is a big proponent of this idea. So ever since reading this book, I've been trying to think, okay, well, if this is right, then there has to be some kind of computation system, some kind of machinery that operates on polynucleotides, but it has to be such that we haven't discovered it yet. So imagining, you can imagine some Turing machine or something at the level of molecules that's operating on DNA, but such a machine would be so complex that it's hard to imagine that we wouldn't have stumbled upon it. This is a Turing machine that operates on DNA would be more complex than, let's say, a ribosome, and ribosomes are visible in electron microscopy. So I haven't seen anything like this. So this is what I'm going to try to propose today, that there is a system that is simple enough that you can imagine it being have gone undetected in the decades of molecular biology, but is also a universal computation system.
So just a silly question: why would it be a lot more complex than a ribosome? This looks a lot like a ribosome to me, I guess, and the jumping around and the, yeah. I don't know.
Yeah, I mean, this is not a very good depiction of a Turing machine. This is just like I guess an illustration. But I guess there are a few things. One is it has to be able to move bidirectionally on DNA. Ribosomes don't do that, and it has to be in a controlled way, not like stochastically, according to some rules. And then the idea is, well, with Turing machines, I guess every Turing machine solves a single problem. But if you want to be able to, achieve universality, you either have to have the capability of, constructing whatever Turing machine you want for different problems, or you can have a universal Turing machine that's able to compute any computable function, and you can just give that function as a string or something. And then the number of rules is kind of, it requires a structure that's a little more elaborate than just mapping codon or triplets of nucleotides to an amino acid. And what the ribosome is doing is pretty simple. It's you take every 3 and map it to a single amino acid and just like construct a string out of that. But to ask a ribosome to do something more complex, whereas like if you see this go into this other state where it changes the rules of what you do based on what you see, and then modify the nucleotide based on what state you're in and what you see, that's a little more complex.
I guess I was thinking of maybe the ribosome reads some sequence, and then that sequence has some effect on the ribosome that maybe moves it somewhere else. Is that not too crazy?
What it constructs has an effect on having it.
Like maybe it translates a protein that then takes the ribosome and puts it in another part of the sequence.
Yeah, maybe. I mean, I don't know if I don't know how. I mean, maybe if you think about that, like, and come up with some formalization that you might be able to show that's a universal computation system. It's I don't know. And I guess I also don't know of any protein that could pick up a ribosome and put it, move it back somewhere.
Yeah, that's true, because the
else.
ribosome recognizes, it recognizes certain sequences.
Yeah, and also you need to modify that the RNA strand that's being transcribed, and that's not part of what the ribosome does.
All right, thanks.
Okay. Yeah. Okay. And then there's also been this debate in genetics regarding the functionality of non-coding RNA, and this is just one paper to kind of I guess link to that body of literature that shows that as you move to larger genome size, the portion of non-coding to coding kind of decreases, non-protein coding portion of the genome. So it's as if the the protein building blocks of an organism kind of saturates and asymptotes at some level, but then something else happens as we get to more complex organisms, complex defined as number of cell types. And so this kind of suggests that maybe there is some undiscovered role of this non-protein coding portion of the genome. And as we, every year, new roles are being discovered for non-coding RNA. This is actually an old paper, but it's like a good depiction of what the concept that
that's really cool. I wish I had that figure in my PhD. Well, I should have seen it. It's from 2012, but I always try to explain this whole non-coding RNA thing.
Yeah,
that would have helped a lot.
Yeah, actually, Chang has also. I think I've seen an updated version of this figure in some other presentation. So it might come up in. It might actually be in one of his later papers, an updated version. I don't know. Yeah. Okay. So now what I'm going to propose is that universal computation is within the reach of nature or evolution at the molecular level. And the way I'm going to show this is I'm going to bridge two very distant concepts, RNA molecular biology and lambda calculus or combinatory logic. And what I'm going to claim is that RNA is astonishingly well suited to run combinatory logic. It's almost hard to believe how perfect it is in so many ways. Okay, and now we'll see that as I go into the details of how to implement a combinatory logic with RNA molecular biology. So first of all, what is combinatory logic? It's the system that actually preceded Turing machines. It can be said to be the first universal computation system in the early 1900s. It consists of these entities that we call combinators. And I'm only going to present combinatory logic. It's a simpler, it's simpler to explain than lambda calculus, I think. So combinatory logic consists of these entities we call combinators, and you start off with a set of primitive combinators and build every other combinator with your set of primitive combinators. So for example, the identity combinator takes a single input and returns that as an output. It's a function that as input is given another combinator and outputs another combinator. It's a system of functions that take functions and return functions. Okay, and then here's some other combinators that implement other rules. And let me just give an example of how to use combinatory logic to do a very simple computation problem. So let's say we want to implement this Boolean logic circuit. The way you do that is you construct this, a string of parenthesized combinators, and then you give as input at the end of the string a bunch of ones and zeros encoded by k and ki, which is like kind of the standard for Boolean combinators. This is Church's encoding for Boolean combinators. And then if you apply these rules step by step, you end up with the answer to this circuit. So for example, I don't know, let's pick a line here, like I don't know, this line where it says k of ki and then of ccki, whatever. To implement this rule, you go to this table here, it says k of x, y equals x. So it says of the two next combinators that follow k, delete the second one, okay? So right here, following k is this thing that's surrounded by parentheses, and then this big thing right here. So to implement k, you have to delete this whole combinator here and also get rid of k because now we're done with it. We just applied it. And then we're left with ki of k and then ki. And you just keep on, mindlessly applying these combinators until you end up with the answer. Is that clear? Yeah, okay. Now, what I've shown here is actually not that impressive because Boolean logic circuits are the weakest of the computation systems that I showed a few slides ago. Remember that, the diagram of different computation systems. Boolean logic circuits are way in the center, the weakest computation system of the ones I showed. But with combinatory logic, you can actually achieve universality. You can, with just a handful of combinators, you can compute any computable function. And there are different systems that do this. So there are different sets of primitive combinators that do this. So one of the famous or most popularized one is the BCKW system. It's these four. These four are enough to compute any computable function. Another is the SK system, or SKI system, sometimes they say. Sometimes they just include this I for convenience. And just with these two, you can compute any computable function. So as an example, you can implement data structures. This is an example of how to implement a pair structure. This is an example of how to implement a stack structure. So a stack with 3 elements, X, Y, and Z, would look something like this term right here, down here. And then you can come up with algorithms that operate on these data structures, even recursive algorithms.
And
so for example,
is the memory recruitment component in there as well? Is it in the kind of copying combinator?
Yes, very good point. Yes, so because we have these copying combinators, for example, W of XY equals XYY, you can increase the length, increase the size of a combinator as you go on, as you proceed through these steps. And so in a way, you can recruit more memory, you can increase the number of states. So you can't capture a combinatory term in a finite dimensional space because one operation might increase its dimensionality, it can add another combinator to it. That's a very good point that you noticed. Yeah. So these examples are 2 example programs that implement recursive algorithms. This top one is a recursive algorithm that removes all the consecutive true elements in a stack and stops when it reaches a false element or the stack is empty. This is another program that reverses all of the elements in a stack. So that stack that I showed you in the previous slide with X, Y, and Z, if you provide that to this program, it'll produce a stack with z, x, z, y, and x in the reverse order. And this will work for a stack of any size.
And I guess if you have a different input to this, you have to rewrite this string, right? There's no distinction between the input and the program here.
What do you mean if you have a different input? Could you clarify?
I guess if there's like the data, I think of the data and the operations, right? And usually the operations can receive any amount of data, but the instructions for executing those operations are the same. But here, if I had a stack of 5 elements that I wanted to pop from versus a stack of 10 elements that I wanted to pop from, I would have to basically write a different string for each of those processes.
No, so that's
Okay.
the point. This actually works for unbounded size, for a stack of
Okay.
any size. The same way that I can write a code in C that would reverse the elements of a stack that someone else provides it with, and it'll reverse the elements of that stack regardless of what the size is. You can do the same thing with combinatory logic.
Okay. Okay.
Yeah, and in general, combinatory logic is as powerful as any functional programming language that I know. It's you can be creative with how you write programs. You can everyone it might have their own style of writing code, and you can write any kind of recursive function that you can with functional programming languages. Okay, so how do we implement this with RNA, as I promised that I will show? So first of all, there's the question of representing these combinators. So RNA or DNA can trivially represent any string, and we already have an example of that with the codon map. We can use triplets of codons to encode amino acids. So if we have this many amino acids, 3 is enough, and you can imagine something like this working for combinatory logic, where maybe a pair or a triplet of nucleotides can be used to represent any of the combinators in your primitive combinator set. Now, the challenge here, and I'm going to talk about this in a couple of slides, is encoding parentheses. So in the combinator terms that I showed you, they consist of primitive combinators, but also parentheses. Now one could say, okay, I'm going to use another code for parenthesis, open parenthesis, and some code for close parenthesis, the same way I guess we do in a computer. But that's not going to be the method that I'm going to use in this computation system for reasons that I will explain. So RNA molecules are frequently edited and modified. And the question becomes, okay, how do I compute after I've assumed that you can represent combinatory logic with RNA molecules? How can I implement these rules with things that we already know to exist, or things that are similar to the editing rules that we know to exist in cells? And the biggest challenge is parenthesis matching. So you know how in the example that I showed, we were confronted with a long stretch of combinators that was surrounded by an open and closed parenthesis, and we had to delete that whole stretch. And in combinatory logic, it becomes necessary to match parentheses. So for example, let's say you're confronted with this open parenthesis. How do you find the closed parenthesis that matches this one? How do you decide what stretch of nucleotides is considered to be a single combinator?
With a stack, no?
Yeah, exactly. With the standard way you do it, and I guess in programming, is with a stack. You move along, you keep track of what your current depth is, and as you move along, you add or subtract depending on whether you saw an open or closed parenthesis until you reach 0. And then once you reach 0, you decide, okay, this is a single term that I have to delete or duplicate or whatever you want. Now this again is something that I think is a little too implausible to assume that for it to occur in cells naturally, because you need something that proceeds along an RNA strand and also keeps track of its depth based on how many times it's encountered, different symbols. And that would require something like a stack, something that has a state of what its current depth is. But fortunately, with RNA, we don't actually have to solve this problem this way, because RNA already solves the problem of parenthesis matching. Now, RNA strands, similar to DNA, can base pair, and they can base pair with themselves. So the same strand can have two different segments of the strand come together and base pair and form these helices. Now, the way that we depict this structure is not with a helix. We just show it with a ladder, but you get the idea. This can be a helix in reality, in three-dimensional world. Now, and the editing rules are very similar to DNA. G pairs with C and A pairs with U, which is equivalent to T. It's the equivalent in RNA. And then you can get very complex secondary structure from just these duplexes. So these are just different examples of transcripts. These are actually real transcripts. I can't remember right now what these are, but one of them is hot air. One of them is yeah, I can't remember what they are. But yeah, these are just examples of endogenous RNA transcripts in the cell, and that we know what their secondary structure is. And you can see these very intricate multiple depths of parentheses in these transcripts, if you call them parentheses. Okay, so this magically solves this problem that I was talking about. You have base pairs come together. You have different segments of the RNA come together and then form a structure that could be interpreted as a open and closed parenthesis to whatever machinery is processing the RNA. You go ahead, Carlos.
I was just going to say, I don't know if I don't think I remember seeing this in the paper, but you for sure know that you can write these structures as a string of parentheses, right? I don't know if that could help like with visualization, because since it really looks a lot like the example of the combinator logic, an RNA secondary structure is just like a bunch of brackets opening and closing. So anyway.
Yeah, that's a good point. I mean, when I was first familiarizing myself with RNA biology, I ran into this and like, oh yeah, well, people know then, and people know that it's parenthesis, but that's, I guess that's just a way of I guess representing what the secondary structure is, but people don't really think of any biological process using it as a parenthesis or interpreting it as a real parenthesis. So this is I guess the main feature of RNA that makes me think that it's astonishingly well suited to implement combinatory logic. And so here are the editing rules that you can use to implement combinatory logic. I'm going to show two sets of editing rules. One is based on left associative combinatory logic. The other one is right associative combinatory logic. And these are just two different parenthesis conventions with just a set of a handful of editing rules. And I just chose these primitive combinators arbitrarily, B, C, F, and W. And just with these 4 combinators and just an additional editing rule that gets rid of leading parentheses, you can compute any computable function. Here's another set of primitive combinators that implement right associative combinatory logic. And this I like more because it doesn't have to be edited at the ends of the strand. It can happen anywhere along the strand. the previous set of editing rules have to happen at the 3' end of the strand, the way that I formalized it. Okay, so this is implementing the B combinator. This is the C combinator. Like the C combinator, I'm just going to go back a few slides just so that you have the list here. Sorry. So the C combinator swaps two elements. It swaps the second and third inputs. So X, Y, and Z becomes X, Z, Y. And so what's the equivalent? Go ahead to here. Okay, so applying the C combinator requires swapping these two elements, Y and Z, to turn it into X, Z, Y. Okay, so this is just one example to give you the idea of what these editing rules would look like in RNA. Okay. Now, okay, so far I've showed you two models. I'm going to show you one more model that is actually, I think, the most plausible one because it doesn't depend on duplicating RNA elements. So these three, B, C, and F, they can be implemented by just cleavage and ligation. So if you, let's say for F, if you just cut the RNA strand here, and then ligate these two ends, you can get Y. And the same with B and C, just with a set of cleaving and ligating at predetermined positions, you can implement these rules. However, for the M combinator or duplicating combinators, as Carlos, you mentioned earlier, they need to duplicate entire segments of the RNA strand, and sometimes these can be very complex long segments. It could include multiple layers of parentheses, and this would require some enzyme or something that does RNA dependent RNA polymerization. And currently we don't think that that happens across all organisms. For at least in humans, we don't know of any RNA dependent RNA polymerase that's endogenously active in humans. But you can actually circumvent this M combinator through a trick that I'll show you in a second. So if you assume that parentheses can be implemented by these RNA duplexes, then it becomes very easy to create an address and reference sequence. So you can get various RNAs operating in the same variable space and being able to interact with one another. You can have a system where some RNA strand is doing some computation, and then the result of that computation is inserted into another RNA for further computation. And it's not that every RNA is its independent program. You can actually have some cohesion in the cell where it's doing some operating in some space of shared memory. The way you do this is you can assume that a variable has an address sequence, and it can be referred to by its reverse complementary sequence. And with a single cleavage and two ligations, you can integrate the content of that variable as a nested term surrounded by open and closed parentheses. Okay, so once you have this, then you have variables. You have names for variables. In combinatory logic, you're not, strictly speaking, you're not allowed to have variables. Everything has to be a primitive combinator or open and closed parentheses or whatever. You can't say, okay, X equals something in terms of Y and Z. But if you relax this constraint, if you can say, okay, I want to define a combinator term in terms of itself, let's say a recursive function in terms of itself, I say X equals some combinator term, and in that term we have another X. Then you actually don't need the duplicating combinator. You don't need M or W, and you can compute any computable function with only, let's say, the B, C, and K combinators. And this is a new system. It's not something that's been proposed before. So I had to, come up with some implementation of a Turing machine, a simulation of a Turing machine based on this system to show that it's universal, and that's in the appendix of the paper. So with this method of addressing, you can get rid of M, and now all of your operations can be done with only cleavage and ligation, and only cleavage and ligation dependent at predetermined positions relative to the motifs. So, all you need is an enzyme that comes in whenever it sees F or something. It decides, okay, I want to cleave here and ligate these two ends. And, there are predetermined positions relative to that motif, the F motif that I recognized. Now, these enzymes could be, other RNA strands. They could be, self-enzymatic RNA elements. But regardless, they're doing something very simple, which is cleaving and ligating at predetermined positions relative to a detected motif.
So oh, sorry. Where you have the F loop, the motif would be defined also in terms of the distance between F and Y, for example. Like that part wouldn't be variable, right?
Yes, yeah, exactly. Or it would have to, if it is variable, it would have to have some way of detecting, okay, what's the base of this RNA duplex? How, go down to the base of this and make a cleavage there or something.
Yeah. Okay, I guess that makes a lot of sense, right? You can imagine a protein just of a certain size binds there, and one side of it clips whatever's on the other side, and it's good to go. Okay, cool.
Yeah. Okay, and just as a proof of principle to show that you can actually do computation, I made this RNA program that does addition, and it does addition of any two arbitrarily large numbers. So if you provide it with numbers as in terms of a stack of digits, and you give that as an input to this program, it can add any two numbers and give you the result as another stack of digits. So for example, if I provide it with this, which is a instruction for adding 2 plus 6, you can see 2 is this part of the RNA, which is literally a 1 and a 0 in a stack, and this is a 1, 1, 0, which these are the binary encodings of 6 and 2. This strand has a address to this RNA. So, something has to come in and insert this RNA at this location based on the base pairing of this and this right here, the address and the reference. And then this RNA has a reference to itself. So this is dependent on perpetual transcription, which is something that we know exists already in cells. So let's say there's a bunch of these transcripts lying around in the cell, transcript A, there's maybe like, 100 copies or something. And then at every point where the program gets to the point where it needs to insert another transcript into itself, it would do so because, there's already some floating around or something. And then this also includes a reference to this other program, just to give you an idea of how, different RNA strands can interact with one another. And this program gets run at the end of the addition to reverse the elements of the stack to correct the computed number or whatever. And if you run this for if you simulate this for various inputs, so 2 plus 6, it requires some number of cleavages and ligations. And if you run this for various inputs, you can see that the number of operations grows logarithmically as a function of the larger number. And the reason why this is important is because combinatory logic has gained a bad reputation for being slow. The most typical way of encoding numbers is a unary system, and unary systems obviously are slow. You do addition; it takes on the order of the size of that number to add two numbers. Whereas I just wanted to show that if you can imagine a binary encoding, and then you can get addition just as efficient as you would on a computer in terms of algorithmic complexity. Okay.
I'm not very familiar with ligations and cleavages, but this is like 10,000, on the order of 10,000 of these operations, right?
Yeah.
How fast, how fast can a cell do that?
Yeah, that's
Like, or alternatively speaking, or another way of saying this, how fast could this cell compute 2 + 6?
Yeah, that's a very good question. A couple points. First of all, so for very simple ligation operations, let's say, for example, if you want to construct an RNA strand off of DNA, the process of transcription, that can happen very fast. That happens at the order of 50 nucleotides per second. So the polymerase that's reading the DNA proceeds at on the order of 50 nucleotides per second. However, this here is, it's a little more complicated than that. You're not reading off of a strand and copying it. An example of something that does cleavage and ligation is the spliceosome, which I guess is when confronted with a newly transcribed RNA strand, it takes out introns from that strand. So it's 2 cleavages and 1 ligation. And that we think happens at the order of 30 seconds. So something like that's going to be a little too slow if you want this to underlie things like cognition or something. However, the point here is it's just a proof of principle. I don't think this is how, what's happening actually inside the cell to, do arithmetics. But it's just a proof of principle that you can construct programs with this system. Maybe someone might come up with some, other clever methods of decreasing the number of operations to do something like addition, or maybe someone can come up with some way of, I guess, coordinating between different cells that would require more parallel computations or less number of cleavages and ligations for something like this.
Yeah, so presumably, if you could reduce the number of cleavages and ligations per cell and parallelize, then you can, of course, do this in much less time.
Yeah. Or if one other possibility that I think we should take seriously is that maybe RNA operations do happen at a very fast time scale, and the spliceosome is not a good example of, or a good estimate of how fast these kind of operations can happen. Because the spliceosome is doing something a little more complicated. It has to detect, motifs that are, pretty far apart. They can be arbitrarily far apart. And it has to bring them together and then do some cleavages and ligations. Here, nothing like that is required. All of the cleavages and ligations are happening at fixed positions relative to the detected motif.
So maybe there's some kind of lower bound that can be estimated based on the association speed of a enzyme to some target.
Yeah, I don't know of a lower bound. I think it's I think an upper bound I think it's reasonable to consider the speed of transcription as an upper bound.
Okay.
So I don't think it can happen faster than, I don't know, actually, I have no idea. This is just like speculation land. Yeah. So, but again, I just want to emphasize that this is just a proof of principle that you can achieve universal computation. You can use a system like this to compute any computable function. And then the question is, how can you get it to optimize and run fast for different kind of things that you want to do.
So I have another question. I don't know if this is leading to your conclusions, but could you take us through maybe an example of a computation that will be done in a cell using something like this? So like, I don't know, take a neuron, I don't know, take any cell you want and just maybe tell me, like, I guess I'm having a hard time connecting this to real world. Like what computation is the cell doing? Because I don't know, maybe it's not computing 2+6, maybe it's computing some concentration of some enzyme in the cell or some protein concentration or some chemical concentration. I don't know. are these the type of computations that you're thinking of, or?
Yeah, so I guess to walk you through example, it's going to be obviously very speculative, and it would require me to presume how this computation system interfaces with the outside world. So, for example, if I explain to you how a computer works, you still don't know how it's going to how you can show something on a screen, how it's going to take input from your keyboard and mouse. And so the question of how this system interfaces with the outside world, is it actually interfacing with neuronal activity? Is there a way to translate activity into RNA and vice versa? Those are, it's a highly, even more speculative territory that I don't know if it's very useful to start speculating in that area. But ultimately, I mean, let's say a more plausible example would be, let's say a cell wants to determine what the plasticity of a certain synapse is. It wants to determine how its weights, a neuron wants to determine how its weights should change from, let's say, getting inputs from upstream neurons and deciding how to add those weights. That can be a computation program that can take a long time. It can take, I don't know, a day or something. And then it can decide based on, some complex program, which one of these weights has to increase, which one has to decrease, or whatever.
I see. And like every experience could be like a run through of the update or something.
Sorry?
Like every experience could be maybe an update of these weights.
Yeah, maybe. If you still want to operate within the synaptic plasticity paradigm of memory, I think though, but like let's say with what Randy Gallistel has been advocating for years now, he thinks that RNA molecules might actually be the engram, or polynucleotides might actually be the memory engram rather than the synaptic plasticity being the engram. And synaptic plasticity could be an expression of the memory or something like that.
What do you mean by engram?
So an engram is the physical substrate that encodes a memory. So there's been this debate, I guess, especially recently in cognitive science and neuroscience, of what is the engram, what is the physical substrate underlying, let's say, a memory of some episodic event or something. And I guess the standard way of thinking about it is that an engram is a set of weights, or some subset of cells with their weights in a network. But some people have been advocating for a different view of cognitive science, where the engram is actually molecules, is specific sequences of polynucleotides. It's a very controversial and radical view, at least at the moment.
Because I guess then those molecules would need some way to interface back, or to be read in some way, right?
Yeah.
Okay. Yeah,
that is one of the many challenges of this theory, although I think it does solve some other challenges that are being overlooked in neuroscience today.
Crazy. I don't know anything about that. Sounds really, really cool.
Yeah. I mean, I think one of the biggest proponents of that idea is Randy Gallistel. If you, I guess, then from his papers and from the stuff that he cites, you can actually find the subset of neuroscientists and psychologists that are taking this idea seriously. Yeah. Okay. So this just to summarize the model. Every RNA molecule is a code that represents a unique function, and to compute the function, to compute the output of a function A applied to input B, the two terms have to be concatenated and evaluated. And evaluation occurs through successive local modifications. One thing that I didn't talk about is that if a strand has multiple editing sites, the order of editing doesn't matter. And this is thanks to some feature that miraculously exists in combinatory logic. It's called the Church-Rosser theorem, where you can have the editing rules happen at any order, and you would end up with the same string. So this allows for parallelization of RNA rules. And each rule has to be implemented by a distinct enzyme or even self-enzymatic RNA elements. And critically, RNA stem loops have to represent parentheses and nested terms. And I talked about addressable memory. And the models do not require complex molecular machinery or anything that remotely resembles the ribosome or something like that. So these can be small. They could have evaded detection through the many decades of molecular biology. And ultimately, universal computation can be achieved with plausible cleavage and ligation operations on RNA. I have a couple extra slides just kind of speculating on the implications of this. I don't know if we have time for me to go through them.
Please.
Yeah, okay. Okay, so I just wanted to point out that these ideas are, they're not completely new. They've been kind of around for a really long time. There was I guess a trend in the 1960s where people were taking molecular engram theories or molecular macromolecular memory theories seriously. So just to kind of represent the ideas of that time, I'm going to read out a quote from Beatrice Gelber in 1962. She was describing a new approach to behavioral problems, which might be called molecular biopsychology. Simply stated, it is the hypothesis that the memory engram must be coded in macromolecules. And I think very an insightful sentence from what she says is possibly the biomechanical and cellular physiological processes which encode a new response are continuous throughout their phyla, as genetic codes are, and therefore would be reasonably similar for a protozoan and a mammal. And very recently, there's been a revival of these ideas. I guess something that's kindled this is some recent evidence, although very controversial, that RNA might actually be underlying some memories. Let's for example, sensitization memory in aplesia, and also this recent study that's has again controversially claimed that the genome or the genome of neurons in prefrontal cortex is edited in various ways, even in healthy patients, not just Alzheimer patients. They looked at both. Okay, so as a conclusion, our current models of biological computation are lacking in computational scope and fall short of Turing equivalence. And just a handful of RNA editing rules is enough to implement combinatorial logic or lambda calculus. The proposed models for computation have identical algorithmic complexity to that of Turing machines and faster than Turing machines if you account for parallel computation, or faster than, I guess, single-headed Turing machines in terms of algorithmic complexity, and universal computation as well within the reach of natural molecular biology. So I'm hoping that even if this idea of RNA-based natural computation is disproved, we still are left with the problem of trying to find life's universal computer at whatever scale it exists. And thank you for listening. Just wanted to thank Gabby Mayman, Jeremy Dittman, Abbas Rizvi, John Maddock, and Randy Galstol, all for their insightful comments, the discussions, and the corrections to the manuscript.
Thanks so much, Hessam. I have to say, without at the risk of sounding corny, that was beautiful work. It's really, really cool. I don't know if we have, do you have time for maybe 5 minutes of questions or something like that, Perry?
Sure, absolutely. Yeah.
I have maybe too detailed a question, but I've seen lately that there's growing interest in the idea of chemical modifications in RNA, where certain enzymes go on to a specific nucleotide and put in a covalent modification, so as a sort of like signal for that specific nucleotide. And that I think then gets recognized by other enzymes and things like that. So I was wondering if you've considered that and if you think it might have some kind of implications for the model.
Yeah, absolutely. I mean, I actually very briefly alluded to this in the manuscript where I said, methylation or other kind of non-standard modifications of nucleotides might have a role in this. I guess yeah, but then I don't have anything interesting to say about how that could help us, I guess find what the motifs are or something. But yeah, I mean, it's very conceivable, I guess, that these are there's some role for nucleotide modifications.
Because I guess one of the limitations you mentioned is the stability of RNA. No? Like usually you think of them as sampling many conformations in time. So I don't know if that places some limits on the kind of capacity of the model, but maybe if the recognition is done at the nucleotide level, while keeping some of the other rules like the nesting, maybe it could add some robustness. I don't know.
Yeah. With the stability, there's also this issue that a lot of the instability, the estimates for the amount of instability of RNA come from studies of messenger RNA, which is encoding for proteins. So both in terms of secondary structure, we think they're less stable, and in terms of the half-life. So non-coding RNA, I think the current estimates are at the range of let's say 7 hours or 6 or 7 hours half-life. And then circular RNA has even longer half-life. So there are ways that these molecules can be stable over long amounts of time.
Any questions, Barry?
Yeah, I have a question actually. Let's say, have you thought about, let's say, an actual experiment that could be done that could maybe, disprove or prove any part of this universal computer in biology? So if you had infinite money, could you say let's do this and we'll answer this question?
Yeah, absolutely. I mean, I've been thinking about this a lot. I think
let's say unbounded. Not
sorry,
infinite.
what's that?
Not infinite, unbounded.
Unbounded money.
Yeah, that's a good one. Yeah, if I had unbounded money, boy, there's a ton of experiments I would do. But faced with the scarcity and funding that limits the kind of experiments that I can propose, I guess. So in terms of falsifiability, I guess there's a few different ways this can be falsified. One is if you show that the non-coding portion of the genome is actually really not important, at least it can be replaced by random sequences, then I guess that's a pretty strong argument against an RNA-based system, because the computation system has to be transcribed from the genome ultimately, or let's say various ways of I guess well, okay, so there are two aspects. One is trying to falsify it. One is trying to find it or prove it. And I'm more interested in proving it or finding the rules. And I think the least least resistant path is to start from sequencing. And my hope is that with advanced sequencing that doesn't discard all the aberrant reads as noise. You might be able to capture 2 snapshots of before and after an editing event, and then that might show you what a rule is. Let's say there's 2 copies of the same transcript being edited and modified just because maybe it's done in parallel for efficiency, and in your RNA data set, you're lucky and you capture the same transcript but at different time points of its computation process. That might reveal some RNA editing rules that haven't been discovered before, and they might be underlying computation. There's other ideas in terms of let's say if you can actually track down an engram in some some I guess animal model. Let's say if we my current work right now is in path integration in fruit flies, and if someone can first of all show that they're path integrating in a very convincing way, and you can localize the memory of the fly's location to, let's say, a single cell or something, then you can try to disrupt the memory by doing some I guess interrogations or affecting RNA composition of the cell or something like that.
Ablations.
Yeah.
So is the question, sorry, I just wanted to follow up, is, because I guess all of these processes that you described, ligation and cleavage, they're already happening in the cell to some extent, but is the question then, can we connect them to some problem?
Well, we want them to not just happen, but happen according to very specific rules. So it's not enough to show that, ligation happens even with some like unanticipated rules, not just through the spliceosome. Like, let's say you find, hey, look, there's some other rule for ligation and cleavage. That's not enough. You have to show that the set of rules that we found constitute a universal computation system. So let's say if I just found something that implements the C combinator without finding the B or the K combinator, that's not enough. And just to clarify, it doesn't have to be these three combinators. There's like an infinite set of universal basis sets for primitive combinators.
Anything else, Terry? All good.
That was it. No, yeah, I think we went through most of what I had. Yeah, thanks so much. I mean, that was the paper was also very well written, and the talk was also very clear. So thanks a lot for taking the time.
Yeah, absolutely. Thank you for having me. My pleasure.
Thanks, Hessam. We really hope to have you back with your next work.
Yeah, keep us posted on any new developments.
All right.
Maybe an experiment or something.
All right. Yeah, hopefully.
All right. See you next time.